I assume it looks like you guys can hear me from my AirPods. It's probably better sound. Everybody can hear you, Cortney. All right, so I'm gonna get out my introduction. So welcome. Thank you, Cortney, for being here. Thank you for taking my message on Instagram, for not knowing me from anybody and just saying, who's this person? And I was like, can you come and speak? I love what you're doing. So thank you for being here without knowing me or anything, and just being somebody that's just such a fan of what you're doing. So, but again, this is for the Metro DC EDS and HSD support group and everybody else that is part of the community. So welcome. Thank you for being here. Submit questions in the Q&A or in the chat. And then once Cortney is done her part of the slides and talking about what she's been doing, and then also a little bit about her story, we will start taking the questions from the Q&A. So first we have Cortney Gensemer. Am I saying that correctly?
Yeah. Yep.
Gensemer. She's a PhD candidate in biomedical science, and will discuss the work at the Medical Center of South Carolina, Norris Lab's research on finding the genetic mutations for hypermobile EDS HEDS. Cortney is interested in common and rare connective tissue disorders. Her current research is focused on genetic causes of HEDS, advancing diagnostic tools to accurately diagnose HEDS, and how to target biological consequences of the disease with therapies. She's also interested in the various comorbidities associated with EDS. Cortney is a fourth-year PhD student from Lancaster, Pennsylvania. She graduated from Westchester University in 2018 with a bachelor's in pharmaceutical product development. After completing her PhD, Cortney hopes to pursue an academic research career. So welcome, you're doing amazing stuff. So congrats, thank you for doing it for us. So fourth year means that you're all like, you're getting to the end, am I right?
Yeah. So the average length of the PhD at our university is around 5 years. So I'm starting my fourth year, but I think I'm getting close.
Well, you're also close to where I first went to school. I went to College of Charleston, and I did aerobics at MUSC like on the daily with my friend Shannon. So I love it down there. I love that you're down there doing this. How ironic. I came back actually after my freshman year and went to Maryland, but you're down there and doing amazing things. So let's leave it to you, and you can just start sharing your slides if you want to. Okay. And also tell us a little bit about you. I know I did the introduction, but a little bit about your journey also.
Yeah, absolutely. Okay, so I'm not great at, can you
see
what
you want to do? Just leave your slides. So you should be able to like have, yeah, so you should be able to kind of press play, you know how they do in PowerPoint.
Okay.
I can see your slides, but you still should be able to press, there should be like kind of a play button, or actually if you go maybe to the top.
Does that work?
I think it might be in PowerPoint. I think if you press like the play button to go through, I'm looking at the bottom of your screen. Do you have a play button? Like it, you know what I mean by a play button? It's like it starts like sliding through the slides. And I think I see it.
Is it working right now?
I see your slides, but
okay.
it's not one at a time. We see the thing at the bottom.
Oh, okay.
So you have to like play the presentation, I think.
Oh, yeah. I know what's happening. I have two monitors and it had them.
There you go.
Okay.
Perfect.
Okay. Awesome. So I'm going to be talking to you a little bit about some of the clinical and scientific discoveries related to HEDS and comorbidities in the Norris Lab. As Kendra said, I am a PhD student in the Norris Lab, and I'll tell you a little bit about our lab. Actually, I believe my research mentor, Dr. Norris, is on here watching right now. So he can feel free to chime in if he wants to about anything. But this is Dr. Norris right here. So that's my research mentor. And here's a photo of our lab. This was taken over the summer when we had our HEDS interns here, which I'll talk about that a little bit too. But the Norris Lab is really a multidisciplinary environment. We have students and postdocs, as well as medical students and undergraduate students that are all working in the lab using various different tools to look at common and rare connective tissue diseases. So Dr. Norris has 25 years of experience understanding the genetics of syndromic and non-syndromic diseases. So when I say syndromic diseases, that's things like Ehlers-Danlos syndrome and Marfan syndrome, and non-syndromic being things like mitral valve prolapse, which our lab has done a lot of research on. And we're really interested in understanding mechanisms that cause altered biology and how that can lead to the development of druggable pathways that can be used to go back into patients. And one of our biggest goals right now, outside of the basic science research we're doing on EDS, is really to develop a center of excellence at MUSC that can bring together both clinical and basic science to provide holistic care for patients with hEDS. So not just research, but also having clinicians in various areas that are important for patient care. And also, if you have questions throughout, feel free to ask me while we're going. I'm totally fine with keeping things super informal, so you don't have to wait until the end. So a little bit about me. I am from Lancaster, Pennsylvania. So some of you from the DC, Maryland area probably think this looks pretty familiar. I grew up in Lancaster, and I went to Westchester University outside of Philadelphia for my undergrad degree. And then I came to MUSC in 2018 after undergrad for my PhD, which is in Charleston, South Carolina, a really awesome city to be doing a PhD in. And I joined the Norris Lab, and this is a photo of myself and Rachel Biggs. She is another PhD student who's now working on hypermobile EDS in the lab. I actually told her she was going to come back to lab tonight, so she might join us for the Q&A at the end. But what a lot of people don't know is I'm also an hEDS patient. So I was diagnosed with hEDS when I was 19 in college, but I had symptoms my entire life. And I was a very competitive athlete for most of my life, which now knowing I have EDS definitely contributed to a lot of the issues I'm still dealing with today. And it's one of the reasons I'm a really huge advocate for earlier diagnosis and having accurate diagnostic tools, because lifestyle changes and earlier intervention with things like physical therapy can absolutely make a huge difference. And then I also just wanted to throw in some pictures of my family because as most of you know, hEDS is genetic, and multiple members of my extended family have hypermobile EDS, including my dad. And my mom has been an awesome support system for all of the medical procedures and surgeries we've gone through. And then my fiance and my dog, who is a perfect emotional support friend. So I don't know what I'd do without my pets and family through all of this. And they've been really, really important and supportive through the entire PhD process while having a chronic illness. I also just wanted to add like a little disclaimer. I'm still a student. I don't have a PhD yet, so I'm not an expert. I will do my best to answer any questions, but now that I know that Dr. Norris is on here, he might be able to elaborate on anything that I don't know. So I'm assuming most of you probably know a good bit about EDS, but I'm just going to give a little background context before I get started. So the Ehlers-Danlos Syndromes are a group of heritable connective tissue disorders. They're characterized by joint hypermobility, skin hyperextensibility, and tissue fragility. And there are currently 14 subtypes of EDS. Some of those involve changes in fibular collagen. You can see the 14 subtypes listed here with the 2017 nomenclature, some previous nomenclatures. I know some physicians still use the type 1, type 2, and type 3 descriptors for types of EDS. Their inheritance patterns, so autosomal dominant or autosomal recessive. And I'll talk a little bit more about what that means later on. And then the genetic basis, so what genes are associated with these types of EDS. And as you'll see here, the hypermobile type of EDS or hEDS does not have a known genetic basis associated with it. And this is something that a lot of people have been working on for many, many years. So hypermobile EDS, it's the most common type of EDS. The exact prevalence is not truly known. 1 in 500 is probably an underestimate. It's a lot more common than a lot of physicians acknowledge, than a lot of people acknowledge in general. Chip likes to use the description of the most common disease you've never heard of. And I think that's really fitting for hypermobile EDS. It involves generalized joint hypermobility and musculoskeletal manifestations due to ligament laxity. And what I like to emphasize is it's not just being double jointed or not being just bendy. It's a lot more than that. It's dislocations and subluxations. It is tendon and ligament tears and ruptures, joint and spinal instability, secondary osteoarthritis, and a lot of chronic pain. And it can affect any joints and often multiple joints in any individual. There is also mild skin involvement in hEDS, so things like stretchy skin, poor wound healing, and abnormal scarring. And then the systemic manifestations, which I think are really important and often ignored. Things like abdominal hernias, pelvic organ prolapse, dental crowding, GI manifestations, which are incredibly common, Chiari malformation, which is shown here, spinal and craniocervical instabilities, mitral valve prolapse, shown right here, and a lot more than what I just listed there. I think something just pops up in the Q&A. I'm going to see if I can find it. Oh, just kidding. Will the presentation be below? Okay, I'll keep
Yeah, I'm following it and answering.
Okay, yeah, how about you tell me if there's anything, because it's hard.
Yeah, don't worry about it. I'm monitoring it.
Okay,
just making sure I know the questions, but yes, all good. You can keep going.
All right. And then hEDS is associated with comorbidities such as postural orthostatic tachycardia syndrome, dysautonomia or POTS, and mast cell activation syndrome. There's also a really big spectrum of severity when it comes to hEDS. You may have patients who need mobility aids or are even sometimes bedbound, and patients who live relatively normal lives with hEDS. What we have noticed is that women typically present more severely affected than men, and the phenotype of hEDS tends to worsen for a lot of people around the time of puberty. And I'll go into a little bit more detail about why we think that's happening later on. So a little bit of genetics 101. I know that a lot of questions were related to genetics that people had pre-submitted. So I just wanted to talk about some concepts that will help make everything else a little bit more clear. And if you have a biology background, then you probably already know what I'm talking about. So genetics in simple terms is just the study of genes, genetic variation, and heredity. So how things are passed down from parent to child. And your DNA is the genetic material in your cells that tell your body how to make things. You can think of your DNA as an instruction manual. So the central dogma of biology is really the concept that DNA is replicated and then transcribed into RNA. And that RNA is then translated to make protein. And protein is the really the material that makes up all of our cells and tissues and organs. When you're looking at EDS, people talk about collagen. Collagen is a protein that supports all different parts of our bodies. And then when we talk about nucleotides, those are the building blocks of DNA. So you have these A's, T's, C's, and G's here. And those are what code your entire genetic sequence. We have our chromosomes, and those are a long piece of DNA, and all of our DNA is organized onto 23 pairs of chromosomes. So different chromosomes have different genes on them. And a gene is simply just a unit of DNA that codes for a protein. So the collagen 1A1 gene codes for collagen 1A1 protein. And each gene has its own specific location on a chromosome. And then one or multiple genes can work together to determine a particular trait. So this example here is showing you a region on a chromosome where a certain gene is that then codes for eye color. And the last part of genetics, don't want to overwhelm you, but the last little piece here I'm going to talk about is more on a mutation level. So an allele is a different form of the same gene. So you get one allele from each parent. And a mutation is when there is a change in your nucleotide sequence. So you might have a C that becomes a T instead. And that means that there's a different allele present in this location. We all have tons and tons and tons of mutations. That doesn't always mean that you have a disease. So a lot of times the context of mutation seems to mean disease, and that's not necessarily true. There's a lot more that has to be done to understand if a mutation would cause a disease. When we talk about genotype, that just means what are the alleles present in that person. So is your genotype there an A and an A? Is your genotype an A and a C? It's going to be those letters that are present in that specific location. And then the word phenotype, which we use a lot when talking about EDS. This is just describing the characteristics of a person. So for example, an individual might have a genotype that causes a specific eye color. The phenotype of that person is that they have blue eyes. So the phenotype of an EDS patient is really specific to their symptoms and comorbidities. And we have a ton of awesome phenotyping data on patients from our registry so far. And the patient phenotype is really, really important for genetic research. It's what helps us figure out how different genes can cause different things. When we talk about heritability and EDS, you see the words autosomal dominant and autosomal recessive used a lot. And a lot of times there are questions where people are asking, what is the chance that I give this to my child? So autosomal dominant means that a single copy of a disease-associated mutation is enough to cause the disease. So here you can see one parent is unaffected, another parent is affected. They only have one copy, but you only need this one blue copy to get the disease. So that means there's a 50% chance that the offspring would have the disease. So here it shows that 2 children got it and 2 children did not. When we talk about autosomal recessive, that means that 2 copies of the disease-associated mutation is needed to cause the disease. And that's when we start to talk about people who might be a carrier. So a carrier parent would have 1 copy of the disease gene, and the other carrier would have another copy. The offspring would then have a 25% chance of having that disease. So you can see this child inherited 2 normal copies. This child got 1 affected copy, so they're a carrier, same thing here. But the affected child received 2 copies. So when you have a recessive disease, you would have to have both parents as carriers of the disease to pass that on. And when we talk about hEDS specifically, hEDS is really believed to be an autosomal dominant condition. In a lot of the studies that have been done so far and looking at pedigrees or family trees, we see that hEDS is inherited in an autosomal dominant fashion, meaning that typically one parent is affected and one or multiple children in that family will also have hEDS. I also wanted to talk a bit about genetic testing. This is something that I see discussed a lot in different support groups, and people have a lot of questions about, should I get genetic testing? What kind of genetic testing should I get? I'm a huge advocate for anyone with any signs of EDS to get genetic testing, even if it's assumed to be hypermobile EDS. If genetic testing is offered to you and it's affordable or it's covered by your insurance, I strongly encourage doing it. The reason why is because many types of EDS will share really similar symptoms and phenotype, and without genetic testing, it's hard to rule out other types for sure. So that being said, I'm going to talk about the different kinds of testing that are out there. So I'm using just a sentence from our lab website as sort of an example here to show what different types of genetic testing will cover. So one example is a SNP microarray. This is looking at pre-selected locations in different genes for DNA changes. So an example of a microarray test would be doing something like 23andMe or Ancestry. And you can see if all of the words here represent genes, these yellow regions are just the specific regions that would be captured on a microarray. I know a lot of people try to look at their 23andMe or Ancestry data for diagnostic information about types of EDS. And the reality is it's going to miss a lot of things. If this was a collagen gene right here and you were looking for information on a type of EDS, you're missing all of these regions of the gene that could potentially be informative. Not to mention that these genetic testing companies are not meant to be diagnostic for any medical condition.
And if I'm not mistaken, I unmuted my mic real quick, but also problem is that sometimes it comes back and it looks like people interpret it as if they have these mutations, and what they don't understand is that they might not necessarily be pathogenic. I mean, I remember one of the stories
Right.
was that somebody had like all the genetic markers for some type of all the types of EDS, and and I was like, no, you'd probably be dead. Like
Yeah.
that doesn't necessarily happen. But I think that can be really confusing and really
Yeah.
hard. So I think I'm glad that you brought that up. So thank you.
Yeah, I on that note, it really is. I mean, it took me a long time to understand how to interpret genetic information correctly. And so I understand that there aren't a lot of doctors out there to help EDS patients, or that genetic testing isn't always accessible. But taking your raw data from these sites and trying to do your own interpretation, I've heard can often lead to a lot of concern and almost hysteria, because I know there's something with 23andMe where it miscalls one particular SNP as vascular EDS when it's not, and it's like a common mutation. And so a lot of people then get very concerned because they're like, oh, do I have vascular EDS, without actually seeing a geneticist. So I strongly encourage trying not to do that and look that up on your own, but really getting a connective tissue panel done, which is what I'm talking about next. So the next thing would be full gene sequencing, or an exon array. So that is a way of looking at specific genes for specific diseases. So the entire gene will be sequenced for known genetic diseases. An example of that would be a connective tissue panel that includes genes for classical EDS, vascular EDS, maybe Marfan syndrome, or osteogenesis imperfecta. It often includes a bunch of genes for connective tissue diseases that might have overlapping symptoms and can help rule out all of the ones that you don't have. And so that is represented here by just these would be the selected genes, and it would sequence that entire gene. But what you'll see is there are still genes and regions that aren't being sequenced. So in order to capture all of that information, that would be doing something called whole exome sequencing. So you can think of all of the words here as the exon of a gene, meaning the coding region. There are these little regions in between that won't be included. Those are known as introns. And there's also little regions in between the letters, actually, that wouldn't be included also. But it gives you a pretty comprehensive overlook of the coding region of your DNA. And then beyond whole exome sequencing is whole genome sequencing. So that includes absolutely everything. Everything's highlighted here. Every single thing, including these introns and non-coding regions, will be sequenced. So what kind of genetic testing is actually going to be helpful for diagnosis? For diagnostic purposes of disease with known genetic mutations, so basically every type of EDS except for hEDS, a connective tissue panel would be appropriate. For research purposes, whole exome or whole genome sequencing is commonly used. And so I'm going to talk about whole exome sequencing a little bit, and how our lab has been using whole exome sequencing to uncover genetic causes of hEDS.
Are biopsies also done along with the testing that you were talking about? If somebody doesn't have a type or they can't figure it out, do they do the biopsies of the skin or something routinely to look at actually what's happening?
To my knowledge, I don't believe biopsies are used anymore. That's something that was done previously before a lot of the genes for types of EDS were identified. I have heard of biopsies being done in other countries, but in the US, that's not typically standard. I'm not familiar with anyone that's currently doing that for diagnostic purposes. For research purposes, that's another story. If you're enrolled in a research study for a biopsy, that would be.
That was more of what I was referring to. If somebody was in a research study and they didn't know what they had, do you do that as well?
Absolutely. There's a lot of great opportunity for research with biopsies. And that's something our lab would like to work with in the future, for sure. So I wanted to put this question in here because this is something that I know people bring up a lot too. So you get a connective tissue panel done by a geneticist, and they say that you have a variant of unknown significance, or a VUS. And people often leave confused or want to know what that means, and if that can help anyone figure out what's going on with them. So a variant of unknown significance basically means that they found a genetic change in one of the genes that they tested, and it's either uncommon or predicted by computer softwares to possibly be damaging in terms of its effect on the protein. Without any additional research into the genetic component there or any downstream biology, there's really no way to confirm that this is causative of EDS, which is why it's unknown significance. It doesn't really jump out as being something they can ignore, but it's not necessarily something that means that it's causing their symptoms either. One of the things that I like to point out is that these panels only include known genes for connective tissue diseases. So a lot of people with hEDS might have a genetic change in another gene that isn't even on one of these panels. So even though it looks like it could be something, there could be other genetic changes that aren't even being picked up by the test that you had done. And one of the things that is unfortunate about these VUSs is that they're really not followed up on. So unfortunately, patients see a clinical geneticist who often spends most of their time seeing patients and doesn't have a research lab. So they'll look at it and they'll say, this is interesting. I don't know what it means. I don't know if it causes your symptoms. And no one really follows up on that biology, which is really unfortunate. And I think that there's a big disconnect between the clinical geneticists and the researchers on that end, because there are a lot of VUSs out there and maybe most of them aren't anything, but it could be explaining some people's symptoms. And until someone follows through with that or publishes on it, it will remain an unknown significance for you and won't really know if it explains your symptoms or not. So I'm going to jump into talking a little bit about what we've been doing, our patient registry, and our research so far. We have actually almost over 2,000 participants that have reached out to enroll in our registry, and we are still consenting patients and collecting samples. So our registry is made up of patients who live in the United States and are over the age of 12 and have a clinical diagnosis of hEDS. Those are the only requirements to be a part of our patient registry. I will have information at the end on how you can enroll in our research, but the goal of our registry is both to collect clinical data, have a list of patients that are interested in being participating in our research, and then to collect saliva samples for genetic research from these patients. So as I said, we have demographics, medical history, and saliva samples from patients. This was, as of last week, we had 549 samples. We have even more than that now. We're getting saliva samples in every single day. But I do have some statistics that I found really interesting from our patient population so far. So the first thing that I wanted to share is the sex and hormonal influence in our patient registry. So, so far, 89% of our patients are female, and we're really interested in this because we see that females often have an increased severity of symptoms and also report an onset of symptoms around the time of puberty, which has really made us interested in how do hormones play a role in connective tissue and in these symptoms that are present in hypermobile EDS. We then looked at family history. So as I talked about hEDS being genetic, we wanted to know what % of our patient registry has a family history of hEDS. And 80% of our participants reported having a family history, which when I talk about our genetics, that will highlight sort of why we've been focusing on hEDS and families and how we can pull genetic information from these families. We also got self-reported Baton scores from patients. So looking at degree of hypermobility, what I thought was really interesting is that absolutely no one reported a score of 3 for some reason. But we have patients all around the spectrum and several patients who report unknown Baton scores. When we looked at joint symptoms and pain, we saw that 62% of participants reported joint dislocations. I think this is really important to highlight because there is often a misconception among clinicians that if you don't dislocate, you don't have EDS. And this really shows that only 62% of our patient registry has reported joint dislocations despite clearly having hypermobile EDS. And 89% do report joint subluxations, and 88% of patients have chronic pain. Looking at specific hEDS phenotypes, so things that are part of the hypermobile EDS diagnostic criteria, we looked at the percentages of different systemic manifestations of the disease. So we saw that mitral valve prolapse and heart valve issues were fairly common. We saw a lot of things in the skin, such as poor wound healing, stretchy skin, and abnormal scarring reaching higher percentages. And then things like abdominal hernias and pelvic organ prolapse also affecting our patient population. But the comorbidities is what I found to be the most interesting. So more than almost 75% of our patients actually report having a confirmed diagnosis of dysautonomia or POTS. Roughly 35% of our patients with mast cell activation syndrome. But the highest comorbidity was gastrointestinal manifestations. 80% of our participants report GI issues. I think this is really telling of an area that needs more research. And from what I've heard from patients, it seems to be one of the most difficult, knowledgeable areas of care to find a physician in. So finding a GI doctor who knows what to do with EDS patients is very difficult. And there's a lot of people out there who need one. We also have recorded things like autism spectrum disorder, sleep disturbances, anxiety and depression were incredibly high. Not surprising given the fact that patients deal with chronic pain, and also that patients are often dismissed or ignored by physicians. ME, CSF, adrenal dysfunction, Raynaud's, CCI, Chiari malformation, and tethered cord were some other things we collected information on. I found it really interesting that things like CCI and Chiari are highly published in EDS, but actually affected a lower percentage of participants than things like Raynaud's, which isn't often published to be associated with hypermobile EDS. So this was really a really cool opportunity for us to really listen to what patients are saying is affecting them, and focus our research towards some of these issues. So we looked into some of our data to see if we could answer some questions. We wanted to know, does Baton score, does the degree of hypermobility as reported by the Baton score correlate with chronic pain? And the answer was no. We found that there was no statistical difference between the two groups, meaning that people with low Baton scores with low degrees of hypermobility had same degree, or if not more degree, of chronic pain than patients that have more hypermobility.
Question about that.
Yeah.
Just thinking of like boys and having, my own boys that are just so stiff, wouldn't necessarily, I don't even know where they fit on the Baton score. I think they're actually, higher than I would think, but do you think that that's part of it too? Is that because they wouldn't necessarily need it?
Wait, say that again. Sorry, it cut out for a second.
Well, I mean, like people that doesn't necessarily, you said it's not necessarily correlating. So is it because there's also people that are missed, like they could have a lower score, but actually be very impacted? I just think of boys that sometimes are very, very stiff.
Yeah.
They wouldn't necessarily, they would often get overlooked.
Yeah, absolutely. I also think that, it's a shame that the current criteria doesn't take into consideration a lot of other joints. I can say personally, I have a ton of hypermobility in my shoulders and my hips. My Baton score is actually not that high. My elbows aren't double jointed. They don't hyperextend, and I have had a lot of surgeries on the joints that are hypermobile. And so I think in general, we're even missing people diagnostically because we're not including a lot of relevant joints.
Right, and hypermobility doesn't equate to flexibility and vice versa. I think a lot of people assume they're one and the same.
Yeah.
Even doctors, so.
Yeah. And then we wanted to know, does the Baton score correlate with the number of comorbidities? So is having a higher hypermobility score correlated with having more comorbidities? And we found that it was somewhat correlated. There was a weak correlation between hypermobility and number of comorbidities. People with a higher Batten score were somewhat more likely to have more comorbidities. But again, the presence of more comorbidities doesn't necessarily mean that their comorbidities are more severe either. There wasn't really any evidence to us that showed that the Batten score was correlated with hardly anything in our patient registry so far. And so I'm going to talk to you a little bit about what we've been working on in the lab in terms of the basic science in this awesome space you can see behind me. A little lab tour. So we have been working on the genetics of hypermobile EDS. And we initially started our studies with a large multi-generational family with hypermobile EDS. So this was a large family with cousins, second cousins, parents, grandparents, that were all affected in every generation. And it allowed us to identify a strong candidate gene for hEDS. The reason that these large families are really beneficial is because you're able to take two people who are distantly related. You can take two second cousins, which is what we did in this case. And we did whole exome sequencing. So we sequenced all of the coding region of all of their genes. And then you can take those two people and compare what they share. So instead of starting with hundreds of thousands of potential causative mutations, started with 48,000, which is still a lot. And that's 48,000 changes that are considered rare. That's why I say, a VUS could be nothing, because we all will have a lot of uncommon changes in our DNA. And so we were able to narrow that 48,000 down to just one gene by sequencing other people in the family and tracking a few top hits from our mutation analysis through that family. So we were able to look at people in the family who didn't have hEDS and confirm that they didn't have that mutation. And then people in the family who did have EDS all shared that same mutation. And I know one of the questions is probably going to be very soon, what is a gene? I'm going to just tell you right now that that's not something we're able to share at this time. It's not because we don't want to. It's because until we're able to publish that information, we are not able to share that publicly. And I just want to say that even if we did share that gene with you right now, it's not going to drastically change the care that you're receiving. So we then took this mutation and we used CRISPR-Cas9, which is a gene editing technology. The people who created CRISPR-Cas9 actually just won a Nobel Prize. It was the second woman in science to ever win a Nobel Prize, which is awesome. And they used that to generate, we used that technology to make a mouse model of disease. So what I mean by that is we used CRISPR-Cas9 to take the mutation we found in that family and put that exact mutation into a mouse. So that mouse is genetically identical to the change that the family had with hypermobile EDS. Now, the reason this is really important is because it gives us a platform to study the mechanism of the disease. I mean, the first step, of course, is to confirm, does the mouse have hypermobile EDS? Does the mouse have other things that are consistent with hypermobile EDS? And that's actually been what's taking us the most time. This biology takes a lot more time than the initial genetics. And all that biology will go into our first publication that we hope to submit soon. But we are looking at things like, how is the connective tissue, how is the collagen different in these mice? And then we can use this mouse to test diagnostic tools and potential therapies. And that's where this model is so important, because without a model of the disease, you're really limited in the science that you're able to do. We also performed whole exome sequencing on individuals and other families for mutations in this gene that we identified, as well as other genes. And so that additional whole exome sequencing information will also be included in our publication. And I really like to use this picture, the cycle of discovery. So kind of an overview of what our goals are. We're starting with patients with hypermobile EDS, and we're using those patients to make genetic discoveries. And we can use those genetic discoveries to inform animal models. So to make models that allow us to study the disease. And that's where the biology comes in. And the biology is the most important part, because without understanding the biology and the mechanisms of how the disease progresses and develops, we would never be able to get to treatments. And so that's our ultimate goal, is to get to a place where we can work on treatments and diagnostic tools for hypermobile EDS and get that back to patients. So as I said, we're submitting our current work for publication really soon. Really soon. We're really excited. We're getting very close. And we're planning on doing a larger scale genetic study this spring to look at genetic changes in over 1,500 patients with hypermobile EDS. So this will really give us a better picture of what is the impact on the larger EDS community. Is this gene a common gene for all people with hypermobile EDS? The answer is going to be no. We anticipate seeing several different genes involved. And how many different genetic changes can we find? And then we're also doing research on comorbidities and how they relate to EDS. So looking at things like POTS and mast cell activation syndrome. And then how hormones can potentially regulate the progression and severity of EDS. And I think this is really important as there are so many women affected with the disease and affected more severely. Understanding how hormones play a role in this connective tissue biology is going to be really important. And then, as I said, looking at the development of diagnostic tools and treatment approaches. But our lab really wants to make a difference in the EDS community outside of just the bench work that we're doing. So outreach, awareness, and advocacy is really important to us. And it's really important to me as a patient with hypermobile EDS too. So one of the things that we did when we first decided to start working on hypermobile EDS in the lab is we published this literature review. This was the first thing I had ever written on the disease, and I wanted it to be a comprehensive review of everything that's published. So starting with, the history of the disease, the prevalence, the symptoms, the comorbidities, the genetics that's out there, which isn't much, and the biology that's been done so far. And we really use it as a way to highlight the need for future genetic research and kind of use it as our stepping stone into the field of EDS research. What's really cool is a few months ago, we heard from the editor of the journal that this literature review is actually the most downloaded article in the journal's history. So it really shows how important this has been for physicians, scientists, and patients in the EDS community.
Congratulations, that's awesome.
Thank you. And then we're trying to be as involved as we can with various societies and interacting with the community. So as we're doing this tonight, EDS Wellness, the Bobby Jones Chiari Syndrome Foundation, the EDS Society, and others. And then promoting awareness and working on fundraising here locally at MUSC. So we recently had an article that was published in the MUSC Catalyst, which is our local MUSC newspaper, about our discovery so far and about Miss America 2020, Camille Schreier, who is an EDS patient and scientist herself. And she came to MUSC to learn more about what we were doing, actually get hands-on in the lab with us, and help use her platform to spread the word about the need for more research and awareness in EDS. And then the last thing that we've been doing, which is really cool and awesome to be a part of, is our Hypermobile EDS intern program. Our intern program was started this summer for the first time, and the goal was really to bring other people with hypermobile EDS into the lab who were undergrads or graduate students with an interest in pursuing a career in research or medicine or healthcare, and have them in the lab and be a part of these discoveries. As patients who have all struggled with being taken seriously, getting proper care and getting diagnosed, it's a really cool opportunity for them to be a part of changing the future for hypermobile EDS. But more than that, we really want to use this as an opportunity to educate them. So not just in the lab, but we had them do various things like shadowing with Dr. Sunil Patel in neurosurgery, being a part of various outreach events and educational things related to EDS so that they go on to their future careers and help train the other physicians, the other genetic counselors, the other PhD scientists, and teach them about EDS. And it's sort of our way of trying to build this greater population of physicians and scientists who understand the disease in the future. You know, it's really hard to change, you know, the old geneticist who doesn't believe in hypermobile EDS and has done this for 40 years. It's really hard to change that guy's opinion. And it's really hard to get that person interested in learning more. But if we start with the future generation of healthcare providers and researchers, I think we have an opportunity to really change how EDS is viewed and how well it's understood, and so people out there know what it is.
That's fantastic.
And
all right,
so,
we
go ahead. No, go ahead.
I was just gonna say, I was thinking that you were getting to the Q&A.
I was about
And you
to.
are.
Yeah.
yeah.
So I just want to leave this up here. If you want to learn more about our research or enroll in our registry, we have a website, thenorrislab.com. There's also a lot of resources that I've tried to put together on there of patient and physician resources. So there's things like CME credits for doctors. There are resources for doctors if you want to take those to your doctors or just doctors you know in general. That is awesome. And there's resources for patients. There is a little survey on our website that will take you to enroll in our registry if you're interested in participating in our research. I do want to say that it might take a little while to hear back if you sign up to be a part of our registry. It could be a few weeks till someone contacts you if you're eligible, but be patient with us. We do have 4 clinical research coordinators who are working on this with us right now. And then applications for our Hypermobile EDS intern program for next summer will open in February of 2022. So if you or anyone you know might be interested, you can feel free to pass along the information about that. And then if you have any additional questions that aren't answered in the Q&A or you think of something later, you can send them to me or Dr. Norris at edsregistry@musc.edu. Oh, and this QR code right here, actually, if you scan that, that will take us to.
It just did. That's so cool.
Yeah.
That's awesome. You guys, I love that. Progressive, modern,
I'm glad
awesome,
it worked.
easy. No, it was like popped right up. It was awesome. Even on the computer screen. It was great. All right, so I will make sure that also I include the information on how you guys can be contacted on the video recording, on the editing, so that it's there and also in the debrief. So I have questions that I can read to you. Can you, when I'm reading and I'm pulling them up, can you just talk a little bit about people that, you know, if they're like, oh, well, I was diagnosed as classical EDS, but wasn't positive for the collagen 5 gene, and they still think I have that, you know, where do those people fall? Can they still apply to be part of your registry or part of the research?
Yeah, so right now, if you have had a connective tissue panel for EDS and it didn't reveal anything in a known EDS gene, and you have a diagnosis, you can absolutely enroll in our research.
Yeah, I thought it was very interesting that you mentioned the skin manifestations that were pretty apparent in a hypermobile EDS. A lot of times that was just always associated with more classical. So that was very interesting to see. Okay, so first somebody said, please tell Cortney that I'm so excited to meet a fellow female Pennsylvania scientist who's interested in genetics and molecular biology. So people are cheering you on.
That's awesome.
Second,
well, I don't know what you do, but if you wanna get a PhD.
She's actually my friend Liz, and she's my saving grace in so many areas, but she is she worked at NIH, and so does her husband.
Oh, awesome.
Yeah, she's amazing. So
there's a lot of scientists that have EDS. Like, it's really cool. I've met 6 other PhD students so far, like in other labs all over the world with EDS, studying biomedical sciences. That's awesome.
Yeah, well, go figure. I met Dr. Pamela Stratton at a yoga retreat, and she studies endometriosis, and it's one of published all over the place. And she had never thought about mast cell stuff or EDS in connection to endometriosis, and she's now doing research on that in the connection. So it's great. Small world. All right. So somebody has said, when would whole genome be appropriate?
So it sort of depends. For diagnostic purposes, that's probably not going to be necessary. I mean, I'm not a clinical geneticist, so there may be another reason why they would want to do whole genome, but for a suspected type of EDS, a connective tissue panel should be fine. Sometimes for research purposes, if you're enrolled in a research study, they might do whole genome. But for a diagnostic purpose, that's not for research, it wouldn't be necessary.
All right. Somebody says, hi, Courtney and Kendra. I'm also a PhD in molecular biology and immunology with hEDS. So hello. And somebody also in the very beginning said, Kendra, who are you? I didn't say who I was. So I apologize. I am Kendra Neilsen Myles. I have been working in the community since I was diagnosed in 2006, after I had my son. And I just started doing all the things more in the community. I worked in the pharmaceutical world, medical device world, started presenting to first-year genetic students at University of Maryland after I was diagnosed, started working and doing more on patient education and physician education because I have a degree in public health. And then I founded EDS Wellness, and then I got into publishing, and here I am. So, and I've had 3 kids since then. So, and then I'm working with the local EDS group to help them do virtual meetings. So that's why I'm connected with them. All right, so somebody said, what aspects of your study do you feel make it particularly strong in the face of science peer review?
So I think our strength is that this is really the first genetic discovery related to hypermobile EDS that's going to have supporting biology. It's not just, we found this mutation and it tracks through a family, and so it probably causes the disease. We have dug deep into the biology. We're understanding how this protein works, how it affects connective tissue, and have a lot of leaders in genetics and connective tissue biology who have been a part of this project. I'm very lucky to have awesome people on my dissertation committee too, who have worked on connective tissue for decades. So I feel very confident that we are doing very strong science. And I'm sure that Chip could probably chime in there too if he wanted to. I don't know if he's still on here.
You can send him a panelist link real quick if he would like. I'm happy to do that, and then he can join because I would love to hear it. But I also agree. I think the way that you have explained the way that you're doing the research is pretty amazing. Does he want a panelist link? He can join. But I say that.
Want to join on video, or are you just watching?
Yeah, I can send him if he just. Yeah, I get his email. I can send him a panelist. I think I can. But I think that's great because you're right. There is families, particularly mine too, that's very, very obvious. But nobody knows I was the first diagnosed in my family. I'm sure I'm not the only one, but it's very, very rampant. Something, whatever it is. And like I said, only one diagnosed.
Well, we often say that. Sorry, go ahead.
He says, I'm here and I think you're doing great.
Okay, so that means he just wants me to keep. He really likes to push the students in the lab to present, and I love it. I mean, it's really cool.
Oh, you're rocking it. I think honestly, this is awesome. I can't thank you enough, both of you, for being here. And I keep thinking, was this lab at NUSC in 1996? How long has that been going on? How long have they been the Norris Lab?
I think he said he started. So he did his PhD here and his postdoc here, and then started his lab here. And I think he said he started his PhD in 95. I might be wrong.
Man, I should have stayed. I loved it there, and I should have. If I would have done the intern program, but this is so great. You guys are doing amazing. All right, so somebody says, if we had a VUS in our connective tissue panels, does your lab want to know about that? And do you want us to provide a sample for you? So somebody who found a VUS,
So if you've had testing and you've had a VUS, but not something that was deemed definitely pathogenic, you can enroll in our study. You can still send in a saliva sample, and you can also share that report with us. So we've had people do that already, where they've after being consented, you can get us that genetic report so we can look at the VUS that was identified.
Awesome. All right. Somebody said, does that mean, you were talking about the Brighton score? I think she means the Baton score. Does that mean that the Baton score as a diagnostic criteria needs to be updated? And I will also say it's a tool to assess generalized joint hypermobility. It's not necessarily a diagnostic criterion of itself, but
unfortunately, there are doctors that I've seen that do use that alone as diagnostic criteria, or don't look at other things. I'm not a clinician and I don't see patients. So I don't know how strong how much weight my opinion on this holds. But I do think that there could be a better tool to assess for hypermobility. I know the reason that they use it is because it's supposed to be a little bit easier to measure. So using joints that you can measure at like a 90 or 180 degree angle where you can actually measure that if you wanted to, makes it a little bit easier. If we're talking shoulder hypermobility, your shoulder and your hip move in so many different directions that like quantitatively having a numeric metric for what is a hypermobile shoulder, what is a hypermobile hip, I know is really difficult to do. So there is a reason behind why they use the joints that they use. I'm not totally sure on the whole hands on the floor thing because that's seems to be more of a test of flexibility than hypermobility. So I definitely think it could be improved. I don't know the improvement that needs to be done though. I don't know what the solution would be.
I will tell you that it requires the spine to be able to extend to be able to put the hand on the floor like that. So it's part flexibility of the hamstrings, but then the spine has to be able to articulate that way to like go past the hips. So you're right though, and how many boys can't do that, or other people can't do that, or they get stiff. So to your point about the other joints, I mean, I think if somebody's assessing somebody, I mean, a lot of people are really, really stiff, and they probably, I mean, how do you even put that into the equation? Okay, so somebody said, where can Canadian hEDS patients be included in your study? Or when can they? Sorry.
You know, I might let Chip see if he has an answer for that. Right now, we are only allowed to enroll patients that are in the United States. I'm not sure if that's going to change. That's not a restriction set by us. That is by the IRB at NUSC. I would love to be able to enroll patients from all over the world, but I'm not sure if that's something that's going to change.
Thank you. I agree. I would love to enroll people from all over the world too, but there's got to be, there's some people doing something in Canada, but I don't know that. Okay.
Oh, Chip just said we're trying to get global, but approvals will likely not be very soon.
Is that, can you explain that is it possible that you can explain approval for like something globally that for people that don't necessarily understand, like why couldn't I just send my DNA?
So any clinical research study anywhere has to be approved by the Institutional Review Board or IRB at that university, at the institution. And so there are a lot of rules and regulations that go into place, and all of that is to protect patients. It doesn't benefit us in any way. It's actually very difficult for us to get things approved. So a lot of things in place to protect patients. And at this time, I don't know if it's because we don't know the right steps to take to be able to enroll patients in other countries. I'm sure there are additional concerns. Every country has their own IRB process and the legal aspects. See, who knows? That's my first IRB I'm a part of, so I don't know.
Well, and also like if you're doing such amazing work and working to get the respect and reputability for doing the science right, I mean, I would want to jeopardize that, you know, but a lot of people were just naive to not understanding the process and why we couldn't just send our saliva to be, you know, assessed.
Yeah, I know there are people who just want to like give me their spit when I'm with them.
Yeah.
Like, no, I have to
like, can I just send it in the mail right now? Seriously, just tell me what I have. Yeah,
we actually got a saliva sample in today. Someone put stickers on it. We do get little cute things sometimes.
I can't say that it hasn't crossed my mind trying to figure out what, you know, I can understand, but yeah, we're all kind of naive to not just understanding. Okay, so somebody said, and I also want to make sure I bring up that email that I forwarded to you so I can read it. And I apologize for my eyesight. I have terrible eyesight thanks to EDS from way back. And I do have my contacts in. All right, somebody said, I recently learned that there's a huge amount of overlap in the genetics between patients with EDS and patients who have been exposed to mold. For me personally, I think mold is the reason why I have more serious symptoms than anybody in my immediate family. Is there research into this connection?
So it's not something that our lab is currently working on. I have heard of things like this where patients that have had mold exposure have similar symptoms and phenotypic overlap, but it's not something our lab is currently focused on at this time.
All right. Somebody is going back to you to being part of your study and submitting. Somebody said you can have a clinical, you said that you needed a clinical diagnosis of hEDS to be included in the study, but it appeared that I think she means Baton scores for many of your submittals have low Baton scores that would not be considered officially hEDS.
Right. So there could be a couple of reasons for that, you know, and I would have to look at patients on an individual level. But one of the reasons could simply be that some of these patients are older. So the Baton score cutoff goes down with age. There is also a 5-point questionnaire that's used to assess history of hypermobility for people with a Baton score that's right below the cutoff. It's also possible that some of these people were diagnosed not as recently, and maybe this is their current Baton score due to injury, aging, stiffness, something like that.
Right, so we're not totally excluded if we were diagnosed with something else or we have a low Baton score. So that's good to know. Somebody asked, once you've published, is there any reason that our current potential hEDS patients couldn't immediately be tested for it if their doctor is willing to order it? Or is there an anticipated wait for the test to become available?
So I mean, if we publish and your doctor contacts a lab and the lab says that they can test for it, if they can somehow have the ability to look at that gene, then I'm sure there would be a way it could be ordered, but it's not going to be on like connective tissue panels right away. For people who might have had whole genome or whole exome sequencing, that's something that your geneticist or genetic counselor could go back and look up that information in the sequencing you had done previously. But in terms of it being added to a connective tissue panel, that can take a lot of time. I mean, and that's not really up to us. It's up to the companies that are doing the testing. So it kind of depends on how quickly they can have the ability to test for that in their lab and get it added onto a panel.
Yeah. Along that same line, somebody said, do you think that the gene that you are working on will be able to be released to more affordable genetic testing labs like Invitae? I think they're talking about similar things, saying that once you have it, do you think it'll be able to be released to be tested for a more affordable genetic testing? Like, I don't know Invitae, but
yeah, so I definitely anticipate that it would be added to panels on those companies for sure.
Right, and it will just take some time.
That process will take.
All right. Somebody said, when you did patient intakes, did you ask about race, ethnicity? You mentioned gender. I'm curious about other demographic data that you may have recorded in your patient population. That's a good question.
Yes, so we have age, race, and ethnicity. We have all of those demographics. I can tell you that the majority of our patients are white. I don't know the exact breakdown in terms of percentages, but you know, there are a lot of different underlying things that could affect that. I know that people of color often don't meet the diagnostic criteria because they might not show scarring or other skin phenotypes as obviously as white patients. They're also less likely to be given a diagnosis just statistically for chronic illness. So I don't know how reflective that information truly is of how the EDS population is actually represented if you had everyone actually had diagnosis.
It's interesting because I remember something, and it was probably a very old, outdated article that was talking about race and ethnicity, and it just had the chance of hypermobility, generalized hypermobility in the specific population. And they actually talked about African Americans actually being quite hypermobile.
Yes.
That was way back, but I just remember one of those very first things talking about hypermobility and prevalence in certain communities.
In terms of like generalized hypermobility, like not EDS, but just reported joint hypermobility, it is actually highest in African populations and Middle Eastern populations. So I think we mentioned that in our literature review, just prevalence of hypermobility in general is higher in those populations.
Interesting, and interesting that that your study has more Caucasian patients, really shows the testament of like who's going to the doctor and who's like looking at these, it's just, I don't know, I think of disparities in healthcare and different things and people wanting to seek help. All right, so somebody's saying, are you planning on using CRISPR to develop treatments and cures?
Is that possible? Maybe. But in terms of treatments and cures, we have other ideas that would be a lot more simple and realistic than CRISPR without giving too much away. There are potentially ways that patients could be treated without needing something like gene editing.
Somebody commented on what we were just talking about in the chat.
Yeah, I saw someone ask if we've been doing specific outreach to increase participation. Significantly high rate of healthcare, racial discrimination.
Yeah. Well, I can say for sure. I mean, that was one of the biggest things we studied in public health, and it's, to this day, it's just so obvious and frustrating and just overt in many ways, just in our face. And how do we change that? It's just, she said that she's been told by most professionals that BIPOC people do not have that. And I don't necessarily know what those refer to, so I apologize.
Do not have EDS, or sorry,
or maybe, yeah, I'm not sure. Are you saying hypermobility or EDS? Whoever asked that question. Correct, EDS. I would say.
Interesting. I've not heard that said before. I've definitely heard the connotation that like it's a disease that affects only white people, which is definitely not true. I know that our entire registry is not white, but I do agree with whoever's. Oh, so I guess there must have been a doctor.
Yeah.
That I've not heard of that. But no, that is not our viewpoint at all. EDS can absolutely affect people of any race or ethnicity, and it's likely that our population is skewed due to some of the things that were mentioned about challenges with race in healthcare.
For sure, and it's just so unfortunate. I'm glad we're talking about this because it's so important for, in so many ways. And I agree with you, it's definitely not correct. Somebody asked, beyond looking at NGS sequencing.
Next generation.
Right. Yeah, sorry. NGS sequencing, sorry. Eyesight and reading. NGS sequencing, has the group also looked into methylation, inked RNA, and promoters as potential impacts?
All right,
Sorry.
We got a scientist in here.
Yeah.
So we have looked a little bit at promoters. That's the only other thing we've really looked at right now. But I think there's a lot of potential to look at other things, both genetically and epigenetically. And I'll let Chip elaborate on that question if he wants to, because he's the smart one.
So if he responds in the chat, maybe you can read it, or he can. All right. Somebody said, you mentioned puberty as a key time frame of worsening symptoms. What, if any, investigation have you observed regarding other hormonal changes, i.e. perimenopause, menopausal, that influence the genetics, or anecdotally, including pregnancy and hysterectomy? Good question.
So far, clinically, we haven't done a huge investigation into hormonal changes, but in terms of in the lab and our animal model, we do know that the protein that is made by the gene we identified is made in the adrenal gland, which is an important hormone producing gland. And so in our mice, we are looking at how hormones play a role in regulating the gene, and how that might affect male and female mice differently. We absolutely are interested though in looking at all of those things. So puberty, menopause, pregnancy, all of that.
Yeah, it's very interesting. Also, it makes me think about people that have like endocrine disorders and problems, and
Okay.
even in my own family. Somebody said regarding what we were talking about, disparity. Somebody said, I've heard about high prevalence of EDS in the ashwagandha. I don't know.
Whoever asked about the genetic stuff, I can't find the question anymore. Chip did just answer that because we are doing GWAS and ATAC-seq as well. And then the high prevalence of EDS in the Ashkenazi Jewish population. I have seen that there are reports of higher incidence of EDS in the Ashkenazi Jewish population. There's also higher reports of a lot of other diseases in general. I don't know that this is something that's been confirmed for hypermobile EDS necessarily. I haven't seen any good studies that really suggest that it is higher in this population. And it's not something we've been really keeping track of in our research.
Brianna Greenspan is a good friend of mine, and her father, she might be a good contact to kind of see if there's any kind of information that she knows of, because her dad owns a genetic testing company. And I know that they had kind of looked into different conditions that affect that community. And then also she has EDS. So she just might be a good point of contact to just know if there's any kind of, and I'm happy to provide her contact information. She's an amazing, amazing advocate and good friend. We are at, I just want to make sure I remind you of the time. It's about 8:13. Are you comfortable going for a little bit longer? There's a couple more questions.
Yeah,
I think we're going
fine.
through them. Is that cool? Okay.
Yeah.
Right. I just want to be cognizant of your time. Somebody asked, what is your confidence level that this gene is hEDS? I think you already touched on that.
Yeah, so I just want to clarify by saying like this gene is hEDS. This gene is not going to explain all of hEDS. There will be other genes. This will be a subset of people with hypermobile EDS. And for a long time, our thing that we used to say was like cautiously optimistic. I'm definitely more than cautiously optimistic now. I'm very confident. Our science has been really solid and really, really cool and interesting. And I'm really excited to get this published soon. We are hoping to publish in a very high impact journal. So we are planning to submit this to publication with Nature in the near future. And the cool part about potentially being in a high impact journal is not only will it really give credibility to the disease, but being in a good journal is going to get it exposed to a lot of physicians and scientists who maybe weren't really thinking about EDS or didn't know about it.
It's great. Well, whatever you guys are doing, I think it's just, I'm astounded and just in awe of like just the depth that you are going and doing the work, and it's just awesome. And thank you, Dr. Norris. Thank you for the lab. Thank you for what you're doing, Cortney. Thank you for continuing. Thank you for being here. I think it's the most exciting, optimistic news and that we've heard in a long time, even, just awesome. So somebody's saying, I have hypermobility and my daughter has hEDS. She has it much worse than me, though we suffer similar symptoms. She is worried that her babies will have the bad syndrome too. The bad symptoms too. Is this cases in your experience? In terms, I guess she's probably referring to the heritability of the severity of the symptoms versus.
Yeah, it's definitely something that we've seen in families with hypermobile EDS. We've seen that the youngest generation is more severely affected than the previous generation. That's also the case in my own family as well. In terms of like knowing why this is happening, I don't know if there's a scientific answer for this. It's not something we've really been able to study in our animal model or anything. But I think it's definitely something that we do see clinically and a real experience for sure.
All right. Somebody's asked, you mentioned potentially simple treatments. Sounds like you can't talk about what that right now. which is fine, but would you be, is there an estimated time frame for the treatment?
Yeah, I don't know if I have an answer to that. Science can be really unpredictable. So, you know, just being able to test things in cells and then in animals and then getting that to people can take years. Obviously, I would like for something like that to happen as quickly as possible, but I've definitely never done this before. This is my first time. So I
Making
don't
a lot
know
of
how,
headway. It's pretty amazing. Awesome.
so I don't know how long something like that would take, but that's definitely something that we're going to be working as hard as we can towards.
Yeah, well, I have no doubt that you guys will keep making waves. Somebody said, would you be able to test an embryo to detect HEDS?
Yeah, I mean, when our genetic information is published, if you were to have your own sequencing done and you knew you had a certain mutation, you could definitely test an embryo for that mutation. I don't know, you know, how you go about doing that. I don't know what kind of doctor does it. I don't know how that's covered by insurance. I don't know any of that information. But like scientifically, it's absolutely something that would be doable if that's something you were interested in.
Right, and once they understand what to look for, I wouldn't see why not.
Yeah.
What are other connections that we can infer since it's produced in the adrenal glands and impact on hormone sleep disturbance? What other connections can we infer since it's produced in the adrenal gland, i.e. the impact of hormones, sleep, and disturbance?
POTS is definitely another thing that you can think about when you start to talk about the adrenal gland, and sleep disturbances. Absolutely.
Do you mean like sleep apnea or just sleep disturbances in general?
So I mean like sleep disturbances having to do with different hormones. In terms of like sleep apnea, I think that's usually believed to be more of a connective tissue obstructive sleep apnea in most patients.
Central is more associated with like the ANS.
Okay.
So
then,
I have a theory on that too.
That could also be explained by things in the adrenal gland for sure. I mean, we don't have any of that data right now, but yeah, you could absolutely think of connections to the adrenal glands.
Okay, somebody's asking a personal question. I'm going to ask this question below, and then I'm going to pull up the email. I think we've gone through most of them, and I want to make sure I pull up that email that was sent. And I had a couple questions. Somebody's saying, are you saying that you expect that the gene that you've identified will be just one of many yet to be identified hEDS genes? From your prior answer, RE confidence level?
Yeah, so definitely of one of many yet to be identified. I like the way you said that, because yet to be identified, because they should be identified and we will hopefully be able to identify them. Yeah, we right now our priority with our publication is focused on the gene we have identified, but that's not like a stopping point for genetics for our lab. We will be doing a lot of other genetic investigation and looking at common genetic changes across the larger EDS population. And the genetic study that we're going to be doing in the spring, known as a genome-wide association study, will reveal a lot more information about genetics on a larger scale of EDS patients.
I have the questions pulled up. I have one side note that I thought about when you talked about looking at the percentage of comorbidities or and presented that they were occurring, like GI issues. Have you also looked at like eye issues, hearing issues? Did you see any of that? Was that part of the questionnaire? Or even physical manifestations like ear deformities or like, you know?
None of that is currently part of our questionnaire, but we can absolutely look at some of those things in our mice. And we have been looking at, you know, our initial goal with our mouse model has been phenotyping the mice and showing that they have things that are consistent with hypermobile EDS. But in that process, we've also been looking at things that are known to be associated with EDS. So we've been looking at the GI tract. We've been looking at, you know, looking for things like mast cells in the skin and various other things. So that's absolutely something that we could look into.
Yeah, well, that's great. Dr. Norris said, yes, there'll be many genes, and it will be interesting to see if certain genes can explain different comorbidities and or severities. I totally agree.
Right.
Um,
So what he means by that is, if we come back with a whole bunch of genes, we can then look at our patient data and see if certain genes correlate. Say one patient has mast cell activation syndrome, hypermobile EDS, and spinal instability, and maybe there's a gene that correlates with that phenotype more than a phenotype that is like POTS and EDS and I don't know, sleep problems or GI manifestations. We might be able to separate out different sort of categories of symptoms with specific genetics down the road.
yeah, I think it's fascinating. It makes me think also of just like, the development in embryo. You mentioned maturation of puberty and hormones and everything. And I just think of like, boys that are developed and they have an undescended testicle at birth and different things that happen during, when they're in development that also kind of signify, who knows, and if there's a correlation to that. The questions that I have here, we already answered one of them, was the genetic testing for embryo. Would a parent necessarily have it in order for the child to have it?
Yeah, so is it necessary? No. Typically, we see hypermobile EDS being inherited from a parent, but there is something called de novo mutations. These are mutations that occur spontaneously. So you could be the first person in your family to have it. And then you could still pass that on to your children, but someone can absolutely be the first. The other thing we commonly see is that it might have been passed through a family through males before it reached a female in the family. So oftentimes the men in the family don't have very severe symptoms, also are like statistically less likely to go to the doctor if something is wrong or to seek answers. So it's not uncommon for a female to be the first diagnosed in a family as well.
Yeah, that's a great point. I'm glad that you brought that up. Along the lines of the question I just asked, they said, at what age is most detectable? Is there an average?
Right. So the reason we've had our cutoff set at the age of 12 is because from the clinical geneticists that we've spoke with, it seems to be pretty agreed upon that under the age of 12, it's hard to truly clinically diagnose hypermobile EDS. Kids tend to naturally be hypermobile, and we start to see changes around puberty, so more obvious signs of the disease. So we don't typically want to enroll children under that age, and a lot of geneticists won't diagnose kids under that age. Now, I know some parents definitely, they know they have EDS and they can see signs in their kid from a very young age, and that's one of the reasons that having a way to either genetically or even, through a blood test diagnose EDS is an important goal of ours, because you could test a child at a very young age and sort of direct them towards lifestyle choices that might lead to better outcomes too.
Right. You mentioned GI issues being so prevalent. Just makes me think of, even my own kids or my history, or just all the people that have had GI symptoms from like way back, from the time they were a baby, just constantly. It just makes you think. So
yeah,
absolutely. I think that we covered everything. If there was, did I miss anything? I don't think I did. So we are literally right about 8:25, and I want to make sure we're conscious of Cortney's time. She's been speaking for a really long time, and the information that she's been able to share is just awesome.
This back up here one more time. Just, can you guys see this?
Yeah.
Just in case anyone didn't get the email or the website before.
And actually, just like I told you, I took a picture of it with my phone just as a screen, and it picked up the QR. I wasn't trying to pick up the QR code, but it did. It just was really fast. It was awesome. I love that you guys are also still taking people for your registry.
Yeah, it's not something that currently has an end date. I know we've had some people say, I'm suspected hypermobile EDS, I'm waiting to see a doctor, or I haven't been diagnosed yet. That's fine. If you get a diagnosis and want to come back and join, we have no end date for our registry.
I love that you have no end date. Dr. Norris said thank you to me for having you on. I'd love to do it again. I have all the questions, and I think that this is so awesome. You answered everything And this is so important. It gives hope, it gives options, it gives just a feeling like, you know, paths are being made, and it's just commendable what you all are doing. And if I could scream it from the rooftops, I would. So I'm trying through this and other ways, and also including, like I mentioned, is it Izzy who does the YouTube channel that's studying genetic counseling? I told you, I love you guys. You're on there, awesome. You're present. So you guys are, you know, sharing reputable information and correcting it if there's any mistakes, and that hasn't been done. And I just love that you guys are there and doing it, and also working and studying in the community to continue to help us. So I just keep cheering you guys on. Whatever I can do, we'll do it again. Do it live, do it on Instagram Live. I mean, just to share the work that you're doing and get support. It's just amazing. Can people come visit the lab if they wanted to? Like, what are their rules? I know this COVID now, but if they're like in Charleston, they're like, hey.
Oh, I should have said that. Absolutely. So for people who are local, local, or if they're just in Charleston, if you want to provide your saliva sample in person, or you want to see the lab while you're here, you totally can. We love doing that. Come say hi. Rachel
I'm coming down. I swear, this is my excuse. Hi, Rachel.
So this is Rachel. She's another PhD student working on EDS in the lab. So I just thought I'd let her say hi real quick. Here to do some late night.
That's awesome. I love what you guys are doing. I love the view of the lab with the, you know, taking a tour of it. I love seeing what you guys are doing. I mean, it really makes you feel like, I mean, you guys are doing the work. It's just awesome. It's not like this out there. It just, you can see what you're doing and what's happening. And you definitely explain it so well and help us understand the process. And I mean, you're making waves, and it's awesome. And thank you, Dr. Norris, for supporting what they're doing in your lab. I wish I had stayed in Charleston, because I definitely would have been a part of it in some way.
Rachel's a College of Charleston grad too.
Yes, I am.
Hey, College of Charleston. It's the best school ever.
So
it was like the best school to go to school with. I go to the beach during the day and lay out and then go to class. It was awesome. If people, your hEDS intern program, can people do virtual internships at this point if they're not local?
It's something we'd be open to having a conversation about. It kind of just depends on what we have available to be done in person versus virtually. I will say that our intern program does include housing allowance and a stipend for the summer. So it's not like an unpaid internship. It is, we will pay you to be here, give you a housing allowance. You get to live in Charleston for the summer. It's pretty cool.
So I assume that they have to be like studying, you know, or could they just want to be doing an internship as part of, you know, job experience? Do they have to be in a current like program?
Yeah, we require that they're currently enrolled in a program with an interest in pursuing a career related to healthcare in EDS.
All right, that's good. I'm glad you clarified that. Dr. Norris
I'm
says you're brilliant. There's one more question, exactly 8:30, that it was just popped up if you're willing to answer it.
What were some of your other surprising findings regarding comorbidities other than frequency? Yeah, talk about mental health. Yeah, I mean, I think I can go back here for a second. I
love those charts. They're awesome.
Thank you. I made them on Canva, actually.
Yeah, it's like so visual and appealing and very like nice, but easy to look at. Way to go.
Chip says the adrenal gland. You know, well, what's interesting is I hadn't initially even considered putting a question about adrenal issues in our like criteria, in our questionnaire for patients. But we had already seen some things that this gene was expressed and made by the adrenal gland. So I was like, oh, like, let's see if anyone has any adrenal issues. And I mean, I thought we'd get like a few people, but it was like roughly like 15% of our patients, which was, that's not something that's often talked about with EDS.
Can you explain adrenal dysfunction, like what qualifies adrenal dysfunction? Because a lot of times it's like kind of a buzzword, but I don't think it's often very clear in terms of what it means or how you qualify for that.
Most patients, it's adrenal insufficiency is what's been reported. There's also with classic like EDS, there is crossover with congenital adrenal hyperplasia too, which is really interesting that there's adrenal relevance in another type of EDS as well. And then also other surprising findings. I think the anxiety and depression was a lot higher than anything that we expected. I expected to see mental health come up because this is a really difficult disease to navigate, both at the doctor's office and in life in general. But I was very surprised to see that, you know, more than half of our patients reported anxiety and depression.
I can't help but wonder how much that correlates to being kind of a highly sensitive person, but also like, you know, one of my sons, he was diagnosed with sensory processing disorder. He's not autistic at 10 months old. It was very clear. So I just can't help but think of people like that, children like that, and how that really just so much goes hand in hand with anxiety and how you feel the world and proprioception. Of course, that's where my brain goes in terms of research too, of like how that impacts. But you're right, it's huge. I could bring a whole boat of my family down, and it's would love to submit their saliva, but it's very, very prevalent, like the anxiety and depression for sure. And you can't make that up in somebody that really super struggles with it versus somebody that doesn't. You can't.
Yeah, and then I also wonder, just with the stigma associated with mental illness, if these numbers are truly higher, and
yeah,
people who just didn't answer yes, that do deal with these problems.
Yeah, well, I can tell you personally, that has been something I've struggled with too. And I've been, as a mom, I've been hell-bent to make sure that we continue to get kids help and not make it the norm in terms of just being part of your life to get help, versus when you really need it and you're feeling down and you feel that stigma and you don't get help. Mental health is huge. So I totally agree with you. I think that's somebody said, I'm a PhD student in clinical psychology at Drexel. Happy to support any future work. Yay. I agree.
Oh, I think it's an area that is so needed. And I don't know anything about psychology. But I think that, you know, the idea of studying how dealing with a chronic illness that is neglected and dismissed by physicians, the impact that has on mental health would be really interesting to look at too.
Dr. Huberman of the Huberman Lab at Stanford is my favorite podcast. He is a neuroscientist and ophthalmologist. He is amazing. And all the things that he talks about make me think of it's not specific to our community, but it just makes you think about so many things, and you learn so much kind of more about yourself and also about what, you know, may play a role in our community. It's just really fascinating, and it's awesome podcast. Somebody said, what about adding urinary bladder issues? That's a good point.
Yeah, that is a good point. That's not something that we currently have in our questionnaire, but it might be worth adding. When I did think about that initially, also, I will say, when we made our questionnaire initially, we thought we were going to get like 50 patients. Like this whole, like, having 2,000 patients in our registry was never something that
we weren't sure what you were doing. You know, you're finally doing this. Dr. Norris is supporting you guys doing the research into this. And like, I mean, it just shows that when you do things right and do it the caliber that you're doing it, I mean, it's awesome.
Yeah.
And yeah.
So I did think that, you know, for a lot of people, those issues are attributed to tethered cord. So I didn't make that a separate question at the time, but it's definitely something that would be worth considering adding to our questionnaire for sure.
All right, well, go to everybody that's currently studying and doing all the things that, you know, maybe someday I'll be able to go back to doing after raising 3 kids, but I live vicariously through you guys. Go.
Never too late.
Totally support you. No, you're right. It's absolutely never too late. And I just love everything that you guys are doing. Thank you, Dr. Norris. Thank you to the people that are on here that are also studying the different things. You guys are making huge waves. And as somebody that was diagnosed so long ago, I love this. This is awesome.
Yeah, and feel free, if you have additional questions, feel free to reach out to us and let us know. Happy to answer anything.
Yep. Thank you guys. Thank you for going over. We'll make sure that we get this recording and we share how to get it connected. Everybody's saying thank you. Dr. Norris says can't do it without you guys. And whatever I can do to help share, we'll do this again. I mean, I love this. This is so important on different levels, and you guys are doing amazing work and deserve all the credit and chance for people to understand what you're doing and support your work. It also helps people feel like there's improvement and work being done to really help our community, and you guys are doing it, and you deserve all the recognition in the world.
Thank you. Thank you so much for having us.
Oh yeah, you're welcome. Nice to see you guys. Everybody says amazing. All right, so hold tight. I'm going to. All right, I'm ending it. Thank you.